Hello boys and girls, this is Mr. So. Welcome back for another lesson of biology. Today we're going through lesson 37 of chapter 28, DNA fingerprinting. Before you start watching the video, remember to like and subscribe my channel and also share the video to your classmates. Let's start. Scientists they found out that for the DNA in every single person, it is very unique, just like the fingerprint on your thumbs. Therefore, scientists they invented the technology called DNA fingerprinting. It helps to identify each of you with an identity. DNA fingerprinting can be very useful in recognizing or checking a person's identity. Not only by checking the person's identity, it can also show the relationships between the child and the biological parents, such as you would like to know whether the child is really biologically related to you. And this scenario usually happens in the TVB series such as溏心風暴啦。經典對白，你唔係長子的孫，你係野仔。Alright, so therefore in a big family, the elderly they suspect that the grandson may not be really biologically related to them， 可能係外面婚外情搞翻嚟嘅，可能執翻嚟嘅。So by using the DNA technology, check DNA 啦 we can know whether the grandson or the son, so-called the son, is it related biologically related to you， 係咪親生㗎啦？In the ancient Chinese royal family， 中國古代皇室啦，咁後宮佳麗3000同皇后梗係互相爭寵，爭咩寵啊？爭皇上嘅寵愛啦，同埋最好就懷有皇上嘅龍種啦，下一代就可以做王子做皇帝啦。咁但係後宮佳麗互相爭鬥，梗係希望鬥快懷有龍種啦。成日都喺度估會唔會人哋個龍種咧係假嘅咧？梗係要陷害佢啦。我哋以前就話要滴血驗親啊，究竟睇下呢個妃子懷有嗰個所謂嘅龍種係咪皇上親生嘅咧？但係而家咧滴血就唔可以驗親嘅，成功驗親嘅方法咧得一種嘅啫，就係我哋而家講緊嘅 DNA fingerprinting 啦，就準確率好高，滴血驗親就非常唔準嘅。呢一課我哋就睇下點樣用 DNA fingerprinting 去驗親啦。Let's take a look at how the DNA fingerprinting works. For 99.9% of the base sequence in a human DNA, they are all the same. However, there is around 0.1% of the DNA which is different between all the human individuals, and this represents the genetic variation between each individual human. And these 0.1% of genetic variation in different human individuals, we can use the technique DNA fingerprinting to identify each of the individual. The drawing below shows you the DNA from mother and the DNA from father. Let's take a look at the 99.9% DNA, which is the same in all human individuals. On the DNA from mother and father, both of them, they have the insulin gene and glucagon gene. This is common in all individuals. And this is the coding region which codes for the protein, insulin, and glucagon, hormone to regulate blood glucose level. And you can find this gene in all human individuals. So it is 99.9% the same in all human DNA. In order to identify a person using the DNA fingerprinting technique, we have to look at the difference and the unique part in an individual, such as in both mother and father DNA, the unique and different part between them is the middle part, the part in black. This part, we call them the non-coding regions, which does not code for any protein. So since it does not code for any useful protein, we call this part in black the junk DNA, 垃圾 DNA. And you can see the length of the junk DNA on mother is longer than that in father. The difference in length of the junk DNA shows the 0.1% genetic variation. It helps to identify a person from another. If you take a look at the detail of each of the small box in the junk DNA, you will find out for each of the small box, it represents some base sequence such as AAA, TTT, CCC, GGG in this way. And this AAA, TTT, CCC, GGG sequence, it does not actually code for protein, and we call them the junk DNA. For the number of junk DNA in both the mother's and father's DNA, it can be slightly different. Let's take a look at the mother's DNA. In the mother's DNA, she has 7 junk DNA as 7 repeats. While in the father's DNA, he has 4 repeats of such junk DNA. But in reality, the number of junk DNA repeats can be any number. So the number I given to you is just an example. So anyway, the number of repeats shows the 0.1% genetic variation between different individuals. And by looking at the number of repeats, which is unique in a person, we can use the DNA fingerprinting to identify a person. Since the amount of junk DNA is unique in different persons and they are very useful in identifying a person, the biologists decided to give these junk DNA repeats a very special name. We call them variable number tandem repeats, in short, VNTR. With different number of the variable number tandem repeats VNTR in different persons, we can use this number to identify different individuals by the DNA fingerprinting. DNA fingerprinting involves 4 major steps. The first step of DNA fingerprinting is to extract DNA. For example, from the blood sample. In forensic science, 鑑證科學, for example, in crime scene, 兇案現場, you can find a lot of blood sample. It might come from the victim, who is dead, or it can come from the murderer, who kills the victim. So in order to know where these blood samples come from, we have to get this blood sample and extract the DNA and identify whether this DNA comes from the murderer or whether the DNA comes from the victim. And after we obtain the DNA, we have to change the DNA into small fragments. But how? After obtaining the very long DNA from the blood stain, we have to cut the DNA into smaller fragments with the help of an enzyme called restriction enzyme. It helps to cut the DNA fragments into smaller pieces with different variable regions. And these variable regions contains the variable number tandem repeats, VNTR. The DNA fragments with different lengths can now be separated according to their length, or what we call the size, using a technique called the gel electrophoresis, 凝膠電泳. Using the technique of gel electrophoresis, some DNA fingerprinting can be produced. As you can see on the gel, there are some DNA bands, and each of the bands consists of DNA fragment of the exact same size. And this particular DNA banding pattern is very unique to every individual. The sample of DNA fragments are then put into the well of the gel slab and get ready to be separated. After that, we will put the gel under the action of an electric field, with the well put into the negatively charged side, while the opposite side put into positive charged. Why should we do that? Because in the well, we have the DNA fragments, and all the DNA, they are negatively charged because of the presence of phosphate group. Phosphate is negatively charged, so all the DNA fragments, they are negatively charged. They will move along the gel from the left to the right, from the negative pole to the positive pole by attraction. However, not all the DNA fragments move with the same speed. The longer DNA fragments, they tend to move slower, while for the shorter DNA fragment, they will move faster and travel further. This is because of the molecular space inside the gel. Inside the gel, it just looks like a sigma, and for the DNA, it is very difficult for them to move along the space or the molecular space inside the gel. If you are very large in size, you are of course moving very slow. But on the other hand, if you are very tiny, you are a very short DNA fragment, you can move through the molecular space of the gel or move through the space in the sigma very quickly. So this is the reason why shorter DNA, they travel faster and easier, while longer DNA, they travel slower. And now the DNA fragments, they are separated into bands according to the size. Staining 染色 allows the DNA bands to be visible, and one very useful technique is to use the fluorescent 熒光. The DNA band become fluorescent 熒光 under the UV light, and you can see them very easily. This picture below shows you how the DNA fragments, they move from the negative pole to the positive pole. And you see there are different DNA bendings because the shorter DNA fragments, they travel faster and further, while the longer DNA fragments, they tend to travel slower and closer to the negative pole. At the very end, the bending patterns from the gel can be copied to the photographic film for long-term storage. And since the DNA fragments we collected from the crime scene, we have no idea whether this blood comes from the murderer or the victim. Using the DNA fingerprinting technique, we can now compare the bending pattern to check whether these blood samples comes from the victim or the murderer.